Hi everyone, it's Mrs. Bustamante. Today we're going to talk about compartmentalization in the cell and why is it really there? Why do we have these different functions within the cell? Why do we have these things called organelles inside this membrane? So I want to take us back and I want us to remember a little bit about cell size. Prokaryotic cells and eukaryotic cells, right, we know are both very small, but when we look at the size of prokaryotic cells compared to that of eukaryotic cells are actually quite a bit bigger than prokaryotic cells than we see in those prokaryotic cells. So we talked about that surface area to volume ratio in cells and how we want it to be big so that nutrients and other materials can pass in and out of the cell pretty easily. And because we even see this difference in prokaryotic cells and eukaryotic cells, right, we need to have some more organelles, some more individual functionality within a cell that allows these eukaryotic cells to be able to function efficiently, right? So when we look at cells themselves, we know that all cells have this one organelle called the plasma membrane. Even prokaryotic bacteria cells have a plasma membrane on them. Both eukaryotic cell types of plant cells and animal cells also have the cell membrane, and we know that it really allows things to enter and exit the cell. What it also does is it kind of just provides a barrier for the cell in order to keep the internal environment of the cell separate from the external environment of the cell, meaning that they can be different. They don't have to be exactly the same, and that cell can undergo specific tasks. Well, eukaryotic cells, though, also have these additional internal membranes in order to kind of help make this process more efficient of moving things in and out and getting things done in the cell. So what are some of these internal organelles, these organelles that are inside of the membrane? We talk about rough and smooth ER, we talk about Golgi complex, Golgi apparatus, right? Lysosomes, mitochondria, chloroplasts. All of these things are examples of organelles that we see inside of our eukaryotic cells that help have a specific job and get things done in the cell. So cellular components of organelles allow for various metabolic processes, or rather tasks, things to be accomplished and transformed in a really simultaneous, meaning they happen all at the same time, way, which increases the efficiency of the cell. That means that the cell can do more than just one thing at one given time. It can have lots of different tasks that get accomplished in simultaneously or even a short amount of time, allowing it to be more efficient for the cell. Lysosomes, for example, right, when we think about what the lysosome does, its job is to lyse or to kill or to break down things, right? Well, the lysosome is a compact little area, right? When we look at it, it looks kind of like a little ball, essentially, right? And inside that little ball of the lysosome are actually hydrolytic enzymes. And they function or they work in an acidic environment, an environment which allows them to break things down. And that's good because then that acidic environment can be within the lysosome instead of the whole cell having to be in an acidic environment, which allows that lysosome to complete the tasks it needs without influencing the whole rest of the cell. So one thing to kind of think about, right, is what would happen if those fluids in the lysosome got released? What if they gave way, they broke free, and they entered the whole cell, right? Well, that would mean that the pH of the cell is changed because those enzymes are there. That acidic environment is released, right? And perhaps it would start killing organelles within the cell, or maybe even the entire cell itself, because the cell is not made to survive in that acidic environment. Hence, having it in that little lysosome, that lysosome having its job is beneficial to the cell. Mitochondria, right? What do we know about the mitochondria? We'll know that the mitochondria have these folds in them, right? And these folds are where we see the electron transport chain happen in the mitochondria, which allows us to synthesize or make lots of ATP in this inner mitochondrial membrane. So just like we talk about surface area and volume with the cells, by having these folds, we get to have a larger surface area in a smaller space, which means that that mitochondria is able to make more ATP because it has more surface area. It has that benefit of allowing it to complete its function more efficiently because it's in a small organelle within the cell. Chloroplasts, right? Chloroplasts also have these folding in the thylakoid that help reactions occur, metabolic reactions occur there, right? They're really folded too to allow the light dependent reactions to happen more quickly, more efficiently, and have more space, more surface area in order to do it, which is beneficial towards the cell. So what happens if any of these things break or don't work, or the loss of these compartments the way they should, right? Well, they're really unique to maintaining the internal environment of the cell. So anytime one of these membrane-bound organelles gets broken or changes in some way or another, it's going to hinder the proper cell function, right? Meaning losing these functions might even cause that cell itself to die if it just can't work properly, or maybe even like we saw in the lysosome, the enzymes in it are released and that causes the cell to die.